हेलो गाइस, वेलकम टू आवर यूट्यूब चैनल एग्रीकल्चर एंड जीके। मेरा नाम है सिंचना और आज मैं आप लोगों से सीड टेक्नोलॉजी में सीड डेवलपमेंट एंड स्ट्रक्चर ऑफ सीड पर लेक्चर डिस्कस करूंगी। वीडियो शुरू करने से पहले एक रिक्वेस्ट है, अगर आप चैनल पर नहीं हैं तो प्लीज़ चैनल को सब्सक्राइब कर लीजिए और इस वीडियो को लाइक और शेयर कर दीजिए। और अगर आप चाहिए तो बेस्ट स्टडी मटेरियल फॉर ऑल कॉम्पिटिटिव एग्जामिनेशंस, सो प्लीज़ जॉइन दिस प्रीमियम व्हाट्सएप ग्रुप। तो चाहिए शुरू करते हैं वीडियो। पहले हम डिस्कस करेंगे सीड डेवलपमेंट एंड मैच्योरेशन। सो हियर इन सीड डेवलपमेंट एंड मैच्योरेशन 5 प्रोसेस बहुत ज़्यादा इंपॉर्टेंट है। पॉलिनेशन, फर्टिलाइजेशन, डेवलपमेंट ऑफ फर्टिलाइज्ड ओविल बाय सेल डिवीजन, accumulation ऑफ रिजर्व फूड मटेरियल एंड लॉस ऑफ मॉइस्चर। सो पहले हम डिस्कस करेंगे pollination. Pollination refers to transfer of pollen grains from one flower to stigma of another flower. ये कहते हैं pollination के लिए. So here next comes to types of pollination. So there are majorly 3 types of pollination, but gytenogamy is another extra type. First coming to self-pollination. Self-pollination or autogamy. Autogamy, it means transfer of pollen grains of the flower into the stigma of the same flower. This is called as self-pollination. Example है paddy, wheat, pulses except pigeon pea. Pigeon pea is often cross-pollinated plants. Next coming to cross-pollination or another name है allogamy. Here transfer of pollen grains of the flower into stigma of different flower on different plant. In cross-pollination, 2 different plants is needed. Or in self-pollination, only एक plant is needed. So here example is maize, bajra and coal crops. Next coming to often cross-pollination. Here in often cross-pollination, ये plant है, ये self-pollinated है. सिर्फ if self-pollination is maximum, but if cross-pollination occurs at the rate of 5 to 30%, then it is called as often cross-pollinated plants. Here example is cotton, sorghum and pigeon pea. Next coming to gytenogamy, another type of pollination. Here एक ही plant needed है, पर इसमें when pollen grains fall onto the stigma of different flower of the same plant, it is called as gytenogamy. ये है types of pollination. चलिए नेक्स्ट स्लाइड पर. नेक्स्ट कमिंग टू फर्टिलाइजेशन. आफ्टर pollination, the pollen is landed on the stigma of the flower and it germinates with the help of the substances secreted by the stigma. Only after stimulation by the stigma, the pollen will germinate. So here when the pollen tube entering the style, it secretes the pectinase enzyme. Pectinase enzyme secrete कर दूंगी pollen, इसीलिए pollen tube grows in the style. So after pollen tube move through the style, it enters to the ovule through 3 different process. ये है porogamy, chalazogamy and mesogamy. When porogamy means when pollen tube enters the ovule through micropylar end, and chalazogamy when pollen tube enters through chalazal end, and mesogamy when pollen tube enters through funiculus or integument. These are the 3 types of pollen tube entry into the ovule, and ये 3 process है, ज़्यादा important है for JRF point of view. And coming to after the pollen tube enters into the embryo sac, the embryo sac consists of embryo sac or a matured ovule or a female gametophyte consists of 8 nucleate or 7 cells. Means there are 7 cells and 8 nucleus. So ये 7 cells कौन सी है? ये 3 cells at micropylar end consisting of 1 egg cell and 2 synergids like this. This is synergids and 1 egg cell. And next 3 antipodal cells at chalazal end. This is chalazal end. Next 1 cell located at the center by fusion of 2 polar nuclei. So this 2 polar nuclei fuse to form a single cell, and this is in diploid condition. 2 polar nuclei, this is in diploid condition, and these one cell and synergids is in haploid, and 3 antipodal cells in haploid condition, and this one cell at the center, that is polar nuclei, is in diploid condition. This is the fertilization. Next, coming to embryo development process. Here, after the pollen tube entering into the ovule, 2 cells is discharged. So 2 gametes is discharged. Okay. So one gamete fuse with egg cell and another gamete fuse with polar nuclei. So this one egg cell fused with egg cell form zygote, and this one egg cell fused with polar nuclei form endosperm. Okay, which is triploid. Coming to zygote. So it's zygote which will be developed into a embryo. So this embryo development process is as follows. So embryo, it is a 2n in condition because of fusion of one egg cell and one male gamete. Okay. So here after zygote, it undergoes mitotic division and form 2 cells. Which is CA and CB. This CA undergoes vertical division and forms 2 cells. Okay, and CB, this undergoes transverse division and forms CI and M. So this will be like this. Here, this is the zygote and it is divided into 2 cells. This is one cell CA and this is CB, and next it undergoes transverse division and one vertical division and transverse division. So after it undergoing this type of division, so here in CA, one cell develop into cotyledon, another cell develop into stem, apex, hypocotyledon, root tip. In case of CB, this CI undergoes transverse division and forms N into N dash suspensor, and this M undergoes vertical division and forms 4 to 6 cells, and this suspensor gives to inner cells which is converted into root apex, and this 4 to 6 cells undergoes and it is converted into outer cells which is called as root cap. So here the zygote which will be divided into 2 cells which will be form a different embryo, that is proembryo, and this proembryo is differentiated into single cotyledon, stem apex, hypocotyledon, root tip, and root apex and root cap, and this forms a complete embryo. So this is the different stages of embryo development. So here it is a pictorial representation. Here this is the embryo sac. Here 3 antipodal cells are there, and here 3 egg cells and one egg cell and 2 synergids, and center polar nuclei. And this zygote formed, it consists of 2 cells CA and CI, and next it undergoes series of division. So here it becomes 8 cell, 16 cell, next early globular shape, late globular shape, and transmission, and later heart shaped embryo, and later completely developed into seedling, that is matured embryo develops here. This is the stages of embryo development. ये है, ज़्यादा इम्पोर्टेंट नहीं है पर इसे नोट कर लीजिए. सिर्फ नेक्स्ट चलिए एंडोस्पर्म डेवलपमेंट. आफ्टर एम्ब्रियो डेवलपमेंट, the polar nuclei which is present in the center, which becomes after fusion with egg cell or after fusion with male gamete, it becomes 3n, and this cell develops into endosperm. So here endosperm development, it develops from the primary endosperm nucleus, and there are 3 types of endosperm development. A है nuclear, cellular and helobial. So where here in the nuclear endosperm development, nucleus undergoes several division before formation of cell wall, like this. Here many nucleus inside a single cell before formation of single cells. But in case of cellular, in there is no nuclear space. That is here 1 cell, 2 cell, 3 cell. This is the cellular type. In the holobial, where the free nuclear division is preceded and followed by cellularization, like this. Here a nucleus form है और a cell है, and this ruptures and forms single cells. This is the holobial type. So nuclear type occurs in wheat, apple, squash, and holobial type occurs in monocots. So remember these points, and ये ज़्यादा important है. Next, coming to structure of seed. So लास्ट लेक्चर पे हम डिस्कस ऑलरेडी कर देंगे. लाइक सीड्स इज कंसिस्ट ऑफ 3 parts: embryo, endosperm and protective coat. And seeds are classified based on the cotyledons as monocots and dicots. So let's see what all this. Coming to components of seeds. Seeds में components है. एक ही पहले है seed coat. It is the outer covering of the seed which gives protection to seed. Okay, and it is developed from the 2 integuments of the ovule. Outer integument develops into testa and inner integument develops into tegmen. ये है सीड कोट. सीड कोट है, ए सीड है और ए सीड कोट है, एंड ये सीड कोट कंसिस्ट ऑफ टेस्टा एंड tegmen either in the fused condition or in separate forms. Okay, next coming to embryo. Embryo, it is the mature ovule which gives rise to the plant. It is the miniature plant which gives rise to the whole plant, and it consists of majorly 3 parts: plumule, radicle and cotyledon. And this plumule is the which gives rise to stem apex here, which is gives rise to vegetative shoot or stem apex, and it is covered by a protective sheath called coleoptile. And radicle, it gives rise to the root apex, and it upon germination, and it covered by a protective coat called coleorhiza, and it also consists of the cotyledon. And based on cotyledon, monocots and dicots. In monocots, only single cotyledon is present, and in dicots 2 cotyledons are present. Next, coming to cotyledons. So cotyledon, these are the compressed seed leaves, means these cotyledons upon germination which develops into 2 seed leaves. So in monocots, it is consist of single cotyledon which is called as scutellum, and in dicots 2 cotyledons are present, and it serves as a storage tissue. So next, coming to endosperm. This develops from primary endosperm nucleus which is formed by fusion of polar nuclei and male gamete. It is a storage organ in the seed. So based on the presence of endosperm, seeds are divided into 2 forms. एक है endosperm or albuminous seed, and non-endospermic and exalbuminous seed. endospermic और albuminous seed में endosperm present है, और इस एग्जाम्पल से कैस्टर बीन एंड फेन्यूग्रीक. बहुत ज़्यादा इंपॉर्टेंट है, नोट कर दीजिए इसको. एंड आफ्टर दैट इन नॉन एंडोस्पर्मिक और एक्साल्बुमिनस सीड, एंडोस्पर्म इज absent, rudimentary or even absent. Here in this case, in non-endospermic seed में cotyledons serve as a storage organ. Cotyledons serve as a storage organ. Example है chickpea, pea and other pulses are non-endospermic or exalbuminous seed. Next, coming to ये है ज़्यादा इम्पॉर्टेंट है, इसमें एग्जाम्पल से बहुत इम्पॉर्टेंट है, नोट कर लीजिए इसको. अपेंडेजेस ऑफ सीड्स. हियर ऑन. सो ऑन है लाइक दिस, ए सीड है, इसमें एक नीडल लाइक शेप से एक प्रोजेक्शन है एट द टिप ऑफ द सीड. इट इज़ कॉल्ड एज ऑन. एग्जाम्पल है पैडी और ओट एंड बार्ली, इसमें ऑन प्रेजेंट है. नेक्स्ट, आफ्टर दिस हाइलम. It is the scar present on the lateral side of the seed. It represents the attachment of the seed stalk to the fruit. ये है seed. पल्सेस के एग्जाम्पल्स, पल्सेस के सीड को लीजिए, और ये है हाइलम. एक स्कार और एज ए पॉइंट इट विल बी लाइक दिस. दिस इज कॉल्ड एज हाइलम. A scar present on the lateral side, on one side of the seed, and it denotes where it is attached to the fruit, or is where it attached to the fruit. फ्रूट के अटैचमेंट के पार्ट को हाइलम देते हैं. नेक्स्ट कमिंग टू स्ट्रोफियोल, इट इज़ द आउटग्रोथ बिलो द हाइलम. सो ये है हाइलम और ये है हाइलम और इसमें एक आउटग्रोथ है, इसे कहते हैं स्ट्रोफियोल. नेक्स्ट कमिंग टू माइक्रोपाइल, द पॉइंट वेयर द इंटेगुमेंट मीट्स एट द न्यूसेलर अपेक्स. सो ये है सीड, ये है सीड, ये है हाइलम और इस रिच को हम कहते हैं माइक्रोपाइल. और वेयर द टू इंटेगुमेंट्स, लाइक दिस इज द हाइलम और where this is ovule, so where these two integuments meet, this point we can call it as micropyle. Next coming to chalazal end, opposite of micropyle end is called as chalazal end. Next coming to rapha, rapha is the area between micropyle and the chalaza, so this between the point is there, no, this is called as rapha. नेक्स्ट कमिंग टू कैरेंकल, ये इम्पॉर्टेंट है. इट इज़ वाइट स्पॉन्जी आउटग्रोथ ऑफ द माइक्रोपाइल सीन इन सम स्पीसीज ऑफ कैस्टर. कैस्टर में ये आउटग्रोथ है, स्पॉन्जी आउटग्रोथ है, इट इज़ कॉल्ड एज कैरेंकल. बहुत इंपॉर्टेंट है JRF पॉइंट ऑफ व्यू. एंड कमिंग टू एरिल्स, इट इज़ ए कलर्ड फ्लैश मास प्रेजेंट ऑन आउटसाइड द सीड. दिस इज सीड एंड दिस इज नटमेग, मीन्स example is nutmeg, so this is the fleshy colored aril. Example is nutmeg. Next coming to hairs, cotton seed example है, cotton seed example hairs which is present on cotton seed. Next coming to wings, so here this is the moringa seed, this is the wings example. So these are the appendages of seed. ज़्यादा से ज़्यादा important है, नोट कर दीजिए इसको. नेक्स्ट कमिंग टू स्ट्रक्चर ऑफ मोनोकॉट सीड और एग्जाम्पल है मेज. मेज सीड को हम एग्जाम्पल देखते हैं. मोनोकॉट सीड्स आर सिंगल सीडेड फ्रूट एंड albuminous in nature. Here endosperm is present. In all monocots endosperm is present and seeds are enclosed in a single seeded fruit called caryopsis. Caryopsis. So example comes to paddy, wheat. For caryopsis example is paddy and wheat. The seed coat is fused with fruit wall called pericarp. This seed coat is fused with the fruit wall called as pericarp. The endosperm forms the major portion of the seed and is separated by an embryo by a layer called epithelium. And its major part in endosperm, the major part is consisting of starch. And outermost layer of endosperm is called as alluron layer. Alluron layer is living tissue. ज़्यादा important है, नोट कर दीजिए. Devoid of galactomannan reserves and secretes the alpha malase enzyme which degrade the endosperm tissue. Alluron layer ज़्यादा important है, ये secrete कर देंगे alpha malase enzyme which is used to degrade the endosperm tissue. If seed is treated with iodine, the endosperm turns black because एक स्टार्च कंटेंट है, इसमें स्टार्च कंटेंट है, इसीलिए ये ब्लैक को टर्न देगा. एंड एम्ब्रियो इनटू येलो कलर. सो ये डिफ्रेंशिएशन हमें दूंगी. एंडोस्पर्म और एम्ब्रियो में आयोडीन को यूज़ कर देंगे और हम टेस्ट कर देंगे एम्ब्रियो कहाँ है और एंडोस्पर्म कहाँ है. ये है द स्ट्रक्चर ऑफ मोनोकॉट सीड. And in monocots, embryo is a single shaped cotyledon, single shaped, that is scutellum, it is present and it is having plumulent radicle which gives rise to shoot apex and radicle in root apex, and this in turn is covered by coleoptile and coleorhiza. And this is a major characteristics of Poaceae and Palmaceae family. Important है, Poaceae Palmaceae family में coleoptile और coleorhiza present है और ये है characteristics of this family seeds. And growing embryo absorbs nutrient from endosperm through epithelium tissue. Note करोगे इसे. And initial synthesis of alpha malase and protease occurs in scutellum. अल्यूरॉन लेयर में अल्फा मेलेस सिंथेसिस होगी, पर इसमें पहले ए अल्फा मेलेस और प्रोटीएस स्कूटेलम में होगी सिंथेसिस फर्स्ट इन स्कूटेलम एंड देन इन अल्यूरॉन लेयर. सो नोट करूँगी, स्कूटेलम इज द सिंगल शील्ड शेप्ड एम्ब्रियो एंड इट कोलियोप्टेल एंड कोलियोराइज इज द कैरेक्टरिस्टिक्स ऑफ पोएसिया एंड पामेसिया फैमिली. And endosperm absorbs nutrients from epithelium tissue and alpha amylase and protease occurs in पहले synthesis occurs in scutellum or after that occurs in the alluron layer. इम्पॉर्टेंट है, नोट कर लीजिए इसको. So here this is the structure of monocot seeds. So here this is the seed coat fused with the fruit wall called as pericarp. Or here this is the major part, endosperm consisting of starch, and this is the epithelium layer. This is the epithelium layer which separates the endosperm from embryo and which helps in the absorption of nutrients from endosperm to embryo. Okay, and here this is separated by alluron layer which secretes the alpha amylase enzyme. And here this is the shield shaped cotyledon, that is this is one single cotyledon, that is scutellum, and this is epicotyle, and this is radical region covered by coleorhiza, and this is coleoptile. So this is the shape of a monocotyledon seed. This is the structure of dicot seed. So here this is the two cotyledons. Okay, this is two cotyledons which consist of storage tissue in case of exalbuminous seed or non-endospermic seed. Okay, and here the seed coat is separate, not fused with the fruit wall. Seed coat is separate, that is testa and tegmen, and it consists of epicotyle and hypocotyle region, and this is the two cotyledons. So this is the structure of dicot seed. Next, coming to structure of dicot seed. Example है Bengal gram. Dicot seed consists of seed coat made up of two layers, that is testa which is thick and brown, tegmen which is thin and whitish, and both may be fused or may be separate. And embryo consists of two fleshy cotyledons along with the embryonic axis. Okay, so here this is the two cotyledon and this is embryonic axis. The embryonic axis consists of radical, hypocotyle and plumule. Okay, two hypocotyle to which the cotyledon, this is hypocotyle to which cotyledons are attached. And here the cotyledons of non-endospermic seed is bulky because it contains the storage tissue, and of endospermic seed is rudimentary or hostorial in nature, like this small or rudimentary. Next, this is an important note, this one. So the nucellus part, in ovule, the nucellus part present in the ovule is after fertilization develops into perisperm in some of the species. Example is coffee and ecca. The nucellus is present as perisperm, that is persistent as perisperm, and this is present in coffee and ecca, but in others it will deteriorate or it will vanish. So this is the important point, note कर लीजिए इसको. सो ये है सीड स्ट्रक्चर और सीड मैच्योरेशन, एम्ब्रियो डेवलपमेंट एक्सेट्रा. सीड मैच्योरेशन में सो आफ्टर द डेवलपमेंट ऑफ द एम्ब्रियो एंड द एंडोस्पर्म एंड ऑल दिस थिंग्स, द सीड विल dry, loses its moisture and reduces its moisture and it become dry, and after that we can say that it will be fully developed, and this is the seed maturation. सो गाइस, आई होप ये वीडियो आपके लिए यूज़फुली होगी. अगर आपको वीडियो अच्छी लगी हो तो प्लीज़ वीडियो को लाइक करें और ज़्यादा से ज़्यादा शेयर करें और हमें सब्सक्राइब करें. थैंक्स फॉर वॉचिंग. मिलेंगे नेक्स्ट वीडियो में.